Thank you both, Dr. Benezra and Dr. Amato. We'll move to the 30-minute QA session now. So please continue to submit your questions and upvote questions that have already been asked. And I'll start with the ones who have been upvoted, and then we'll go in chronological order after that. So let's see. The first question looks like it may actually go back to Dr. Amato. It's a question from Ken Lassen. And the question is, there are over a dozen studies on microbiome shifts seen with long COVID. Are you aware of any microbiome intervention studies for this condition?
I am not aware of specific interventions that have been put forth thus far, although this is not my area of research, so there could be things out there I'm just not aware of. But I do know there's been a lot of interest, not only in long COVID, but also in terms of like severity of illness, in terms of thinking about, cytokine storms and this kind of runaway inflammation that you get and people that get really severe cases of COVID, and how that could be linked to essentially the body's propensity to kind of move towards inflammation, and the microbiome could be contributing to that, as well as contributing to some of the underlying conditions we know that increase risk of severe COVID, like obesity, diabetes, things like that I talked about in terms of being disparities. So I think there's thoughts about how can we kind of promote an anti-inflammatory microbiome more generally in terms of maybe diet or probiotics, things like that. But I don't know that we have a kind of silver bullet solution, which is true for all of microbiome research, essentially.
That makes sense. The next question was actually the most upvoted question for Dr. Benezra. The question was, do the participants get any insights back about their microbiomes? But I might couch that in a broader question of, how do you pitch a study? How do you recruit families into a study when there's such a different understanding of what you're looking for, and how do you explain that, and how do they benefit from this?
Yeah, that's a great question. So I can talk specifically about the work I was involved in the Gordon Lab, but in general, the concept surrounding microbiome is a very high income country concept. It's not a priority for people who are, battling with infectious disease or like constant enteric disease. In some ways, it's a luxury science, not to insult anyone here, honestly, but, it's a concept. So in the study that I was working in, all of the Bengali field research assistants, and I asked them specifically about this, did not talk about good microbes, microbiome that is important to your health. They only talked about pathogens. So the focus was only on hygiene, clean water, disease-causing organisms, because there was a feeling that it would confuse the issue too much if we started talking about commensals and good microbes and all this other stuff, which is really problematic, right? Because that's all that the Gordon Lab scientists cared about, was thinking about beneficial, I don't even know the right words to use, because as all of you as microbiome experts know, that there's no such thing as a good or bad microbe, right? It depends on the time and place and, all kinds of other surrounding factors. But there was absolutely no understanding that microbes could be important to your health. It was deliberately left out of the conversation. So there was never going to be any parity in terms of understanding what the science or the study was about. But on the other hand, something I often think about is all the children in that study lived, which is not true of everyone in that community. So that is a really important outcome, and was the mother's priority, obviously. Like, they really didn't care about the science. They cared about keeping their kids alive. So how to figure out how to introduce concepts that will be useful or valuable, what is the benefit sharing of microbiome science for these populations, is a very good question that not really very many people are tackling or taking on.
Thank you. Next question, let's see. Again, for Dr. Benezra, this is from Christian Diner. What is your vision on unifying anthropological factors with the microbiome field that always tries to represent individuals by incomplete sets of quantifications, metadata that rarely describe the complexity of those factors?
Yeah, that's that's like my life's work to try to figure out the answer to that question. I think just having these conversations like we're having right now, having scientists and microbiome experts think about that this stuff might matter too, even though it's really hard to quantify and messy and is also very frustrating on my end, right? Like many times I've given talks to scientists who have asked me to quantify my data. They're like, well, put it in some kind of understandable set for us. And I can't, and I refuse to, I won't, because I did talk to 9 families. I can tell you 8 out of 9 families use toilet paper. I mean, is that a useful quantity for you? No, probably not, right? But the point of ethnography is that it's giving this very granular, everyday ethnographic picture of life that may be useful to you in how you think about your metagenomic data sets or something. Like there have to be ways where we have to learn how to talk to each other because we really do speak different languages now. And have different priorities. Again, like Dr. Gordon and I both really felt strongly about developing a working interdisciplinary collaboration, and we still couldn't really figure it out. So not to be like a downer that it's not possible, because I feel like it's worth continuing to work on. But those kinds of data just don't even transact. But maybe we can make space for some more kind of ethnographic information in the scientific data, right? We can think very critically about how we're using categories of race. We can think about what are more precise ways to define populations, right? What are their toxic exposures? What are their diets? What are their all kinds of other social, biological, environmental conditions, instead of saying, this is what I always come back to thinking about like vaginal microbiome studies. The Revell study says Black women have these microbes, white women have these microbes, Asian women have these microbes. That's a very damaging way of thinking about microbiomes, right?
Thank you. The next question is in the same realm. It was asked of Dr. Amato, is by Christian Diner. I feel like many of the questionnaires used in microbiome studies do not capture determinants of health or inequities well. What do you think needs to change in order to correctly quantify the environment that we are sampling from?
Yeah, I mean, I think this is very similar to what Dr. Benezra was just saying. It's difficult, right? I agree that they don't quantify it effectively. I think first and foremost, and this goes back to some of what Dr. Benezra has written about, right? But why are we putting a certain variable into the study? Is that really the variable we want? Is it really race or is it racism? Because it's probably racism, and in that sense, we should be measuring racism, and that can be experienced in a lot of different ways, right? And so I think trying to be very deliberate about how we're designing surveys and why. And I think this goes back to kind of, from my perspective, kind of a basic foundation of science is that we should have like a priori hypothesis about relationships between variables that could help guide some of that. And I think because the technology for being able to look at these microbiomes emerged so rapidly, and there was so much excitement in terms of just trying to describe these communities, that at the beginning a lot of that kind of fell by the wayside, in terms of people just wanting to describe what they could find and kind of saying, this came from this place and that came from that place, and they're different, right? Because they eat different diets, but also like really there's so many things that are different about those two places. And so maybe you only collected those diet data because that's what you could do, because there wasn't like this a priori hypothesis, because we had no idea what the relationships were going to be at the beginning, right? But I think now that we're starting to have an idea of like, these are some of the things kind of at a broad scale that are influencing the microbiome, to try to kind of go in and be a lot more deliberate in what we're measuring and why. And so, I mean, it's still difficult, right? We are never going to be able to capture that complexity entirely. But, you know, if you're focusing on diet, making sure you're measuring diet carefully and being explicit about what part of diet you're interested in and why, right? I think that's the best we can do kind of on the scientific side, and also engage in these discussions that Dr. Benezra was talking about in terms of, you know, here's what I'm thinking, but what's all the other stuff that's happening that I haven't even considered? And is there a way we could quantify that or not? And I'm a little different because I'm a, you know, scientist in an anthropology department, but I feel very strongly that like in my lab, if people are working on a data set, to the extent that we're able to make it happen, I want people to go visit that field site, because even though it's not the kind of detailed ethnography that Dr. Benezra does, at least that's some sort of kind of qualitative input people can get that will help them interpret the data, right? Maybe it's not getting written into the results, but at least people can then say, here's the patterns we saw, here's the categories that it aligns with, but like, here's all the other stuff that could be happening in terms of like diet or you know, access to water, that we couldn't really quantify, but is probably playing a role, right? Or like, here's why that point might be out here. Like, I know that family was different and I couldn't really quantify why, but like, it makes sense that that family landed over here and this family landed over there. So I don't know if that was a great straight answer, because it is a hard thing, but that's what we try to do anyway.
And you touched a bit in your answer. Oh, Dr. Benezra, did you have something to say? No.
I was just going to interject something, but I don't want to.
Go ahead.
Just, I think that a really good example of what Dr. Amato is talking about is that, so again, the Gordon Lab is really interested in making probiotics, and we all want to solve the problem of childhood malnutrition, right? There, we can agree that that's the goal, to like try to fix that problem. They're really focused on a probiotic intervention, right? So I was always like this annoying voice being like, but people are still going to have open drains in front of their houses. They're still going to have pit latrines and terrible sanitation and no like government run infrastructural services. So you can give them lots of probiotics, but those structural problems are still going to exist. And so then their health problems are still going to exist, right? So just getting people to think about those as components of the problem, not as separate, right? Like not as peripheral, but as contributing to the problem is my goal.
Yeah, and who's going to make the probiotics and who's going to pay for it also are my questions.
And that actually segues well into the next question, which was asked by Dr. Gibbons about policy, which you did touch a little bit on a little later in your talk when you talked about, and this is for Dr. Amato, though you're both welcome to chime in. But specifically when you were talking about, maternity leave and how can we facilitate things from a policy level. But his specific question, if there's ways to elaborate, how might we craft policy to target and mitigate known microbiome adjacent inequities in marginalized communities? What would be the most impactful thing to do first?
That's a great question. I mean, my knee-jerk reaction is starting with some of the kind of early life parental leave interventions, because I think that's where a lot of these disparities in microbial exposure is set up, and then is perpetuated. So I think, making sure that people have the infrastructure they need to be able to have a healthy pregnancy and increased chances for vaginal delivery, breastfeeding, that they can take time to think about the nutritional input their kids are getting and kind of what it means versus, just making sure that they're fed, water quality, all of those things. I think targeting kind of that early life period would be a good starting point. In some ways, I also go to that because there's a lot of kind of policy that's trying to target that area to begin with, right? And then the trick becomes kind of what does microbiome research add to that, right? Like we already know that parental leave policies in the US are terrible. And so why do we need microbiome data for that? And, you know, we know that breastfeeding is good. And when people can do it, they should do it. Why do we need microbiome data for that? But I do think there are aspects of microbiome research that can kind of add on to that, right? In terms of like, with breastfeeding, I know there's people thinking about like, how different is it to like actually breastfeed versus give an infant pumped milk from a bottle, right? They're still getting the nutritional input of breast milk, but what happens in the microbial community when it's kept in a refrigerator and then reheated? We know that there are like daily fluctuations in what breast milk looks like. So does it, you know, how much does that influence the infant nutritionally and microbially if you're giving the infant, you know, milk that was pumped in the morning at night? You know, what is the infant getting by being like actually in contact with mom physically versus being fed from a bottle by somebody else? Those are things that I think can be can enrich perhaps attempts at improving policy that is currently, you know, either in existence or being developed. So that's where I would tend to go. Although there are so many things that could be fixed about environmental disparity, it's kind of hard to pick one. You know, I think food deserts would be another, you know, access to high fiber, like fresh produce would be another place I would go immediately, since diet has such a strong impact on the microbiome.
And you even mentioned in your talk the circadian rhythms and shift work and policies that could cause protections in that as well. So.
Yeah, no, so many things, you know, parks. How are we building parks? There's all these new fancy parks being made out of like the rubber stuff and metal, right? But like maybe sand and gravel is better because it's an environmental exposure, right? So, you know, yes, we're building parks, but how are we building them? Out of what materials, and how does that affect microbial exposure? So yeah, there's a lot of different directions, but I guess where I would go first would be early life, since that's when the microbiome is getting established. And you're gonna like set up disparities for sure at that point. I don't know.
Thank you. The next question is for Dr. Benezra from Dr. Gibbons. Thanks for that beautiful talk. I think it's critical to understand how we can create fruitful collaborations between social scientists, anthropologists, and biologists. What were the biggest impediments for making this work well in your experience?
So many. But I don't want to sound like discouraged or not hopeful. I think that the kinds of solutions that we each want to come up with. So when I teach my students about this kind of thing, we talk often about Paul Farmer's ideas of proximal and distal interventions, and that they don't have to be mutually exclusive, right? You can think about structural things and immediate interventions as simultaneous and so often, like, especially global health has a really specific intention and way of doing things. And social scientists, I mean, I'd be the first to admit, we can be super obnoxious and really critical, like we're the ethics police and we just are like really good at pointing out what science is doing wrong. And that is not helpful either, right? So figuring out how to work together and be like a willingness and generosity is extreme. I mean, collaborations are all about individuals, right? They're about like individual people trying to work together. So I think that so for me personally, the biggest impediments were that when I started working at the Gordon Lab, I was a PhD student, right? I was a PhD candidate, and Dr. Gordon, as you all know, is the father of microbiome. So there was a huge power disparity that was never going to be resolved, understandably, right? And so the ways in which, you know, science wants to solve problems and anthropologists want to solve problems, and what those problems are really difficult to resolve with one another. And so that is really, and how to talk about the science and what the priorities are really, really difficult. And again, let's be honest, science funding is always going to be more than social science funding, right? There's always going to be a power and money dynamic there. And I think that we have to just keep trying to talk through what is actually important But getting scientists to value qualitative data is probably the hardest thing that I have to do. To be like, this is not just something nice for you to know about, that this is affecting microbiomes in a really important way, and will make your science better and will make it more effective and more precise. And so that can be frustrating for social scientists, and then we just give up. So social scientists need to keep going as well. But I've been doing this work for a long time, and it's very hard for me to get funded because there are a lot of disciplines are institutionalized in terms of journals, in terms of grants, in terms of jobs, right? You can't be an interdisciplinary free-for-all. There's no faculty positions that are like interdisciplinary free-for-all, right? You have to be in a discipline to get a tenure track job, or to publish in a journal. So, places like M Systems are thinking more about who's publishing in their journal. Like all of this equity stuff also has to do with who's doing the science, not only who are the scientific subjects, but who's doing the science, who are the editors, who are the reviewers. All of that is also super important. So as those boundaries start to break down a little bit, it becomes easier, but anthropology organizations never want to fund me. They're like, that's science. You're way too sciencey. But also scientific organizations don't want to fund me because they're like, what are you talking about? That's nonsense. So, where does that leave me? Hopefully invited to symposiums like this, which is great. But I don't know if that answers Sean's question at all, but
it does. Thank you. Next question is for Dr. Amato. Can you please discuss a little bit about the complexity of diet assessments that include underrepresented populations and how this may affect microbiome research? So I feel like both of you have had experience with this, if you'd like to add.
Yeah, I'll start by saying, Katie, as you said at the beginning, I started in field primatology, right? That's my initial background, and looking at feeding ecology and diet and the microbiome in that context. And a lot of the kind of general principles and relationships I'm interested in I think are transferable to humans, although humans are much more complex in terms of lifestyle variation. And so I struggle with this question all the time because it's so much easier to get data from wild primates. People are like, how do you do that? I'm like, well, I do have to go out in the forest and follow them around, but once I'm there, I literally follow them around all day and write down every single thing they ate, and I pick it up and I can analyze what's in it. And that's harder to do with people. It's possible to do, but you can't do it with very many people, and it's super invasive, right? People don't want you following them around all day necessarily, like writing everything down they do. And so, we rely on surveys often. And I think there's so many potential problems with these surveys, right? In terms of, like I said, what part of diet are we interested in? So a lot of times these dietary differences are being reduced down to like macronutrient differences or fiber differences. And it's something that I've done also, because at a certain point you have to just decide how you're going to categorize diets. And even if you have a super detailed dietary survey, you can't put like 100 different questions about diet into a quantitative spreadsheet and get something out of it, right? Maybe one person eats this, one person eats that, and I can't say anything about that, even if I think it might matter. So I really struggle with how to best do the dietary data. There's also questions, sorry, I could talk for hours about diet. Are we worried about like 24-hour dietary recalls, like what the person ate in the past 24 hours, or kind of what their general pattern is overall? Like, usually I eat this, usually I eat that. It seems to me like often the latter is more related with patterns that we see in microbiome data if we're going to have to categorize and quantify. But that also leaves a lot to be desired in terms of kind of detail here. And in terms of like the spices that Dr. Benezra was talking about and kind of how food is prepared, like that gets left out a lot, right? It's like protein, lipids, carbohydrates, fiber. But I think it matters if people are making that in their house, if they're making it with oil, if they're going out, right? And we try to capture these things in surveys, but it's always going to be imperfect. So again, I think I would go back to, I think it's currently oversimplified. I think it will always be oversimplified because it has to be at some level to quantify it from the kind of sciencey perspective. But I think making sure that you have clear hypotheses about what are the relationships you're interested in, right? Are you interested in fiber for a particular reason? And what are the other things that could be interacting with that? So that's kind of my best attempt at dealing with that. But I know there's been people also exploring, I'll just say briefly, beyond looking at macronutrients, looking at like metabolite profiles of the food, or kind of trying to get like more detailed. And in some respects, that's getting past the foods, and in some ways could be more standardized, but I still think you run into the issue of like how it was prepared. I'm not sure the extent to which that would come out in like metabolite profiles. So yeah, I'm not sure I don't think anybody has a good answer, but that's how we try to deal with it.
Dr. Benezra, would you like to elaborate on how to actually study humans in fieldwork? You actually have done that.
Yes, that's true, but I didn't write down everything they ate every second of the day. And I know that when I was in St. Louis, they tried to do a twin study where they had people stay in like a hotel room for 5 days so that they could control their diet. And I think half of the people dropped. They were like, fuck this. Like they were like getting paid thousands of dollars and they were like, no, thank you. I can't do it. Right. Human studies are so difficult in that way. But I think that what Dr. Amato is saying, in just even like accounting for, it's not just maybe components of, nutritional components or like the biochemistry of the food, but also all the other things that go in DACA, families share cooking pots and also share kitchens with 5 other families, it's going to have an effect on the food that they are cooking, and obviously the microbes as well. And so, yeah, the diet surveillance in the Bangladesh study was done by the field research assistants, and it was like a food recall. Like, what did you have for the last 24 hours? Which obviously people are terrible at remembering things, as we all are. Like, I could not tell you what I ate in the last 24 hours. So maybe thinking about how important is that precise, detailed information, right? Like, what other kinds of things might it be more useful to know? I don't know.
Thank you. I think we have time for one last question. There's a question that applies to both of you. It was asked of Dr. Benezra. Do you have any advice for quantitative scientists wanting to collaborate with social scientists, or vice versa?
I mean, it's awesome that people want to do that. And I guess thinking about because I think part of the problem is that when you're trained as a microbiome scientist, you're not trained in all of these things, right? No one expects you to have expertise, just to seek out expertise of people who might have a doctorate in studying humans and culture and things like that, right? So I guess being aware of what you don't know and seeking out an expert who might know it, right? And just accounting for these things as important, right? Because there are anthropologists who are experts in every single country on Earth, right? And every region of every country on Earth, and could tell you a lot about the very specific diets of people in very specific places. But yeah, we never like connect with those people, or microbiome scientists aren't thinking about that. So I think just even asking that question about who else could I add to my team, right? Or my research design, or thinking when I'm writing a grant, I mean, anthropologists are super cheap. We really do not, we don't have labs. We don't have a lot of specialized equipment. We have like notebooks and tape recorders, and we're like very inexpensive. So, next time you're writing a grant, think about, how can I very cheaply add a social scientist that will enrich my data in these ways, right?
Thank you.
Yeah, I would second that. And I think the other thing that I would add in terms of I haven't had a collaboration over like a long-term project, but I've done little kind of papers or panels here and there. I think the point that Dr. Benezra made earlier about like open-mindedness and also understanding that the language is really different, right? So reaching out to people about collaborating, but then realizing that it might take a little bit for you to actually understand each other. And then the other thing that was brought to my attention and I've kind of been thinking about is also like, what do each of you need out of the project, right? Because I know there's also sometimes, I think, although Dr. Benezra, you can correct me if I'm wrong, there's sometimes a hesitancy maybe from some of the social cultural anthropologists in terms of, you don't want to just be like the ethics police or the like, here's how you define this, right? Like you have your own research goals and your own, kind of career milestones, right? That you have to, that you have your own vested interest in things. And that might not always be the same as it is for a quantitative research scientist on the other side. And so making sure you can both get something valuable out of it, I think is also an important discussion to have, so that, it doesn't feel like one person is kind of taking advantage of the other necessarily. I don't know.
Great. Well, thank you. We're out of time now, so I'll go ahead and conclude our Q&A session with another warm thank you to Dr. Amato and Benezra for giving us such a grand finale to our symposium today, as well as extend another round of virtual thanks to all of our fantastic speakers. And with that, I'll return the floor to Dr. Gibbons for closing remarks.
Thank you.
Thank you.
Thanks.